Okay, next week your assignment will include making a model. Your model has to be done on paper and hand written and drawn. I'm not looking for fancy artists. I am looking for accurate information. It cannot be printed, it cannot be downloaded. This is for originality. I want to make sure that you're able to illustrate what you have learned about protein synthesis and mutations. So it has to be done by hand to be graded. So there's two parts to it. The first part is you need to show that you understand protein synthesis. So you need to go from the DNA strand to the mRNA strand to the protein that's made that gives the trait. So within that model, you must include the nucleus, you must include the original DNA strand, you must show the base pairs and how they match up using the ACTG code. You must show where transcription and translation happen and how it happens. So that means you need to include the nucleus of the cell and the cytoplasm of the cell. So this has to be illustrated once. The second part is you need to make a gene code that codes for a trait. Now that will be your normal trait. Then you need to mutate that same trait 3 times, illustrating that one mutation is neutral, meaning it has the same benefits in the environment as the original gene allele. And then you must mutate it, meaning you change the DNA code. So you are making this up, but what's expressed is what you're changing. It has to be the same trait, but expressed different ways. So you have to have one that's beneficial, meaning that it survives better in the environment, one that's neutral, survives the same in the environment, and another allele that causes harm, that it survives worse in the environment. So you will illustrate this by changing the code and showing and drawing the different traits. So part one, you're going to have to show, there's many ways you can do it. You're going to have to show DNA being transcribed to mRNA in the nucleus. You see it says nucleus here. Then the mRNA, you have to show it leaving the nucleus to go to the cytoplasm to be translated into the amino acid strand, which is the protein, which gives us our trait. So at its simplest form, DNA to mRNA is transcribed, but you must show this happening in the nucleus. The mRNA leaves the nucleus, goes to the cytoplasm, protein is made, and that leads to your trait. So these are two examples that I'm giving you, but this is what part one is. Part two is you're going to have to make up a DNA strand of at least 4 codons or 4 triplets, and then you're going to have to show the normal trait. Then you're going to have to show the mRNA then you're going to have to show it being what it looks like normally, create a mutation, either a substitution, an insertion, or a deletion mutation. And then you're going to have to show how it's interpreted. So you have your DNA, your mRNA, you're going to have your normal protein, and you're going to tell me what that trait codes for. Then you're going to have your neutral one, where you show some sort of difference in the code with the letters, and it's going to show that it has the same benefits in the environment. Then you're going to have a loss of function one, which would be harmful, meaning that it doesn't survive as well. And then a beneficial one, meaning it survives better. All of these different mutations have to be of the same gene, meaning it's expressing the same trait. For instance, feathers, red versus brown versus blue. Wings, short versus long versus pointy versus fat and stubby. So they're all describing the same trait. All right, so here's some examples of past students. So this person started off with their double-stranded DNA, which is represented by the pink pipe cleaner. Then the blue came in and copied it. That's representing the RNA. They have their code for which one of the beads represents each of the bases. They showed their original DNA strand being transcribed into the mRNA being translated into the protein used over here. And then they start up here with their normal gene, their 3 mutated genes, and showing what each of those traits are. In this example, it's feather color and why it's harmful, why it's beneficial, why it's neutral. This person did extra. They showed the protein synthesis process for each of the different alleles, which you only have to show protein synthesis once. You need to show your code and your mutations for each one. So this part that I'm showing, that I'm circling with the pointer here, from the code down, you have to do for your different mutations. So they labeled it harmful, neutral, normal, and beneficial. And they also show their transcription, but they don't have the cytoplasm labeled here, so they lost points on that. So they show transcription and translation. They don't have the protein up here, but they have it here. So make sure if you just draw it once that you go all the way from DNA strand to the protein that is there to the trait. Here's another one. They did transcription in one box, translation in another, and then different gene expressions here. Same thing here. They started with the DNA sequence, but they didn't do at least 4 triplets. They only did 1 here. And make sure you start with the TAC on the DNA TAC code, which goes to AUG, which gives you the start and stop. So you need to go from start to stop in your gene code. And here again is some different types of mutations. So this would be part 1, and this would be part 2. And then the last part, after you're finished with it, you're going to take your model and you're going to explain, for example, if this was your model. Like my mutation that I chose is butterfly wing size. No one can do butterfly wing size since I am giving this as an example. So my normal gene was average size wings. But then a mutation occurred where G was replaced by an A. So this was substitution mutation. So I still ended up with average wings, but the mutation did occur and it's a neutral mutation. This mutation caused the butterflies to have a smaller wing, which meant they couldn't fly as well and get away from their predators. So that was a harmful mutation. Whereas this mutation changed the genetic code and allowed them to have bigger wings, which allowed them to get away from their predators. So I'm making all this up, just using the language of DNA to show that I understand how a mutation can change the genetic variation within a species for a gene that expresses a trait, that codes for a trait that's expressed. All right, so hope that helps. And the last part I did, that's what you're going to have to make a video on. No, your face does not have to be in the video, but I do need you to speak in the video. Okay, so that is what you will be working on next week.